Good afternoon, everyone. Thank you for joining me in this talk, which I hope will give you an overview of the next solutions that Acronym is developing and providing to the cell and gene therapy field. And I will use this presentation to put some focus on the solutions that we are bringing to the Treg therapies. So Acronym started back in 2006 as a premier provider of GMP cytokines and growth factors to enable the development of advanced therapies. And being testimony of the exponential growth of the industry and its needs towards commercialization, the company has embraced the challenge, adopting new products and systems that support industrial scale, fully GMP compliant and automated production. So along that road, Acronym has always tried to be a pioneer in the industry, launching several products that have represented different inflection points in the field, like the virus-inactivated, chino-free human blood-derived products, or the first liquid GMP IL-2 in the market for ex vivo use. Acronym's acquisition by Artline back in 2019 enabled the acceleration of such growth to keep being in the front line, anticipating the needs of the industry. And the proof of that are the multiple milestones that have been reached in almost only 2 years, like the electronic drug master files that we have filed with the FDA for IL-2, 7, 15, and 21, the new GMP plasmid facility that Acronym has built, or the addition of our newest member of the family, the viral inactivated AAV serum that I will present in a bit. And one of the milestones that we are most proud of is the first commercialized drug that has been supported by one of our electronic DMFs. So during these years, listening to the needs of this field, we have developed a portfolio that can be summarized in these 4 main blocks: cytokines and growth factors, plasma-derived supplements, custom media transfection and electroporation buffers, and plasmid DNA manufacturing. As I mentioned, Acronym started in the business of cytokines and growth factors production, and as the field has been refining and demanding top quality raw and ancillary materials, Acronym has focused its attention on meeting these expectations, offering fully GMP, highly pure materials. As the industry keeps expanding, we realize how important it is to bring solutions that support the commercial scale and the regulatory compliance. And examples of that are our fully GMP large-scale manufacturing using pharma-grade purification methods that result in cleaner and more pure cytokines, also being the first with electronic DMFs covering IL-2, 7, 15, and 21, or the available cytokines in liquid-filled syringes for ex vivo use. And as we are aware that the field is moving towards functionally closed systems, we have leveraged our many years of expertise on the liquid formulation for IL-2 to also optimize and bring IL-7, 15, and 21 in liquid formats with 24 months of stability, which will be available in single-use weldable bags very soon, similar to the one that you can see here in the image. Acronym has over the years been focused on the immunotherapy field, building an exclusive catalog that has supported mainly T-cell therapies. But as the field has been expanding and exploring other cell types, Acronym has incorporated new cytokines and growth factors demanded by other therapies like NK or pluripotent stem cell therapies. To demonstrate our commitment with the field, we have run several studies to show the equivalence between Acronym's product and some of other ones available in the market. For example, this study that you can see here was made in collaboration with Dana-Farber Cancer Institute. And what you can see here is the equivalence between Acronym's IL-2 and Proleukin. Nowadays, one of the bottlenecks that the industry is facing and that due to the current situation is more present than ever, is securing the ancillary material supply. So it is critical to have multiple validated suppliers in order to mitigate any disruption in the process development and manufacturing. We must take into account that at the end of the day, it is the patients' lives that are at stake. Consistency and reproducibility of cytokine activity will strongly affect manufacturing costs, timelines, and overall quality of cellular therapies. In order to have multiple validated suppliers, one has to make sure that the same product from different vendors has equivalent performance. This is especially difficult when comparing cytokines from different manufacturers due to two main reasons. One is that some manufacturers have a huge batch-to-batch variability, and the other one is that today there is no standardized analytical procedure to determine biological activity and a specific activity for the cytokines, resulting in inconsistent results when using the reported activities. So these two factors make it very difficult to switch from one supplier to another, something that is more needed every day to secure the process. And secondly, here, since at Acronym we want to make things easier for our customers, we have invested some resources to find a solution. So first, you can see here that we have a consistent trend in the activity among different batches. And secondly, we have run several empirical studies having different manufacturers and a standard side by side, measuring the potency with a parallel line approach, which is a currently used method by the World Health Organization for the international standards. And these studies allow us to normalize and find a correction factor that would allow a direct comparison between suppliers' activity levels. Another group of products that Acronym has been expanding over the years is the human blood-derived products as supplement medias. And as human-derived, these supplements present another challenge, which is safety. Especially these days that we are living in with a global pandemic, it is crucial as a provider to ensure a reliable supply of safe product. And the result of this commitment is the recent launch of our newest human AV serum, jointly developed under the exclusive agreement with Octapharma. This product starts from Octaplus, an FDA-approved pooled plasma from US donors. It is manufactured under ISO 13485, and the larger pools that are used in this product offer a very high batch-to-batch consistency. But the main feature that makes this product revolutionary and outstanding is that since the starting material is Octaplus, the resulting AV serum is virus inactivated. Another virus inactivated product that we have in our portfolio is the human fibronectin solution, which also counts with an excellent safety profile thanks to the pharma-grade purification process used in its manufacturing, again with an electronic drug master file available. On the AV serum, it is the compilation of many steps along the manufacturing process that achieves the viral inactivation of enveloped and non-enveloped viruses, conferring the required safety and reliability. So, in summary, Acronym, together with Octapharma, has developed a revolutionary product that is not only safe and compliant with the regulations, but also with a great performance and backed by a highly robust supply chain. Another supplement media that has been optimized for commercial manufacturing and that puts Acronym in the front line is the human serum albumin. 5. The donors have been tested per approved guidelines and collected from FDA licensed donor centers. And since it doesn't contain stabilizers, it has been optimized for cell therapy further manufacturing. It is GMP manufactured in an ISO-certified facility, and it is supported by an electronic DMF as well. Unlike the cytokines, Acronym is also offering the HSA in weldable bags to suit functionally closed systems, providing a very versatile packaging. We will actually have the first batch of HSA in bags in the coming months. At Acronym, we know that in the cell and gene therapy field, there is no one-fits-all solution. So we also offer custom services to help on the development of exclusive pieces that will contribute to distinctive therapies. We have many years of expertise on the customized manufacture of recombinant proteins, medias, and transfection agents from custom media development, formulation, and testing to the largest scale manufacturing, because we want to work with all sizes of companies, from the biggest ones that require a commercial scale to academic groups that are starting their journey towards an IND filing. At Acronym, we keep listening to our customers and the industry needs to keep innovating and onboarding new products that with the highest GMP quality that comply with the main regulatory agencies. And we like to see ourselves as a partner of your process that is willing to adapt so we can accompany you from the beginning towards commercialization. So in summary, Acronym continues to optimize ancillary materials to address key bottlenecks and support the industrialization of cell and gene therapy production. How we do that? With functional closure of critical ancillary materials through innovation and liquid formulation and primary packaging, with larger scale manufacturing to meet commercial demand in recombinant proteins, blood-derived products, and cryopreservation solutions, prioritizing safety and consistency with virus-inactivated human blood-derived products, and fostering resilient advanced therapy supply chains through a geographically dispersed network of manufacturing facilities. And with that, thank you very much for your attention. Any question, please feel free to contact me during the conference or through these email addresses, and you are more than welcome to pass by our booth. Thank you very much.
All right, great. Thanks, Sara. We have a couple questions from the audience. I have one myself. So one of the audience questions are, what about cryopreservation of cells without DMSO?
So this is a very good question. Yes, we are indeed developing DMSO-free cryomedias at this point. It's undergoing. So we have collaborations with several partners, and we are working on that. So yeah, we will keep you posted. Excellent.
And I have a question regarding CMC regulatory filing, especially in the EMA after Brexit. Have the regulations changed at all between accessing the UK market and the rest of the EU, or are they following the same CMC principles?
For now, we don't have like any update on any change. We keep supplying customers in UK as well as the rest of Europe. So we have not noticed any change on that matter.
And do you know what the largest volume size fermenter you would have for cell-based therapies or cell lines? I mean, are these up to 1,000 liters, or are they small scale? And where can you deliver? Is it up to phase 3 or only phase 1?
But do you mean like for media manufacturing, something like that?
Yes.
Yeah, no. We can perfectly accommodate commercial scale up to 200 liters without any problem.
Okay. All right.
Yeah. So
excellent.
phase 3, it's perfectly fine and going towards commercial as well.
Okay. All right. Good. I think, Rochelle, there's no other questions here in the docket. Thank you, Sara, for your time. Appreciate presenting.